Hello, I'm Dr. Vernon Reichenbaker. I'm here from Marshall University in Huntington, West Virginia, and I'll be talking with you about some aspects of medical genetics. In medical genetics, we're going to be talking about topics like single gene diseases, that is, those genetic diseases caused by simple mutations in one single gene. We'll talk about how those diseases are inherited within individual families. And we'll also talk about how those diseases are inherited in large populations. We'll talk about cytogenetics, that is, big changes in chromosomes that can cause problems. We'll talk about what we're beginning to learn about the inheritance of relatively common diseases in the population, heart disease, cancer, and that sort of thing. We'll also talk about gene mapping, how we can identify the location of genes and the distance between genes, and we'll learn how that gene mapping information is useful in genetic testing, genetic diagnosis, and the cloning of genes. Okay. As much as possible, what I intend to do is to focus on the material that is high yield for the USMLE Step 1 exam. And let's get started with Chapter 1, which concerns the topic of single gene diseases. Single gene diseases are those genetic diseases caused by simple mutations just in one single gene. And in this chapter, we'll look at how those diseases are inherited in individual families by analyzing pedigrees. We'll also look at some complications that makes the analysis of those pedigrees difficult sometimes. Let's begin by looking at some basic definitions, just so that we're sure that that everyone knows the basic vocabulary that we'll be using in this topic. The first term is gene. Gene is the basic unit of inheritance. It's what gets passed along from generation to generation to determine most of our characteristics. Genes are made of DNA. Genes can play a variety of roles in the cell. They can play regulatory roles and so forth. But the most important kind of gene for us in medical genetics are genes that code for the structure of proteins. Because one of the general rules of medical genetics is that almost always when there is a mutation that causes a genetic disease, almost always that mutation occurs in a gene that codes for the structure of a protein. The term locus refers to the physical location of a gene. That is, exactly where is that piece of DNA located? Is it on chromosome number 10? Exactly where on chromosome number 10 is it found? All of our genes are located in particular places on particular chromosomes. Humans have about 30,000 genes, and each of those genes has its own particular locus or physical location on a particular chromosome. We use the term allele when there are two or more variations of the same gene. For example, if we were discussing sickle cell disease, in our discussion of sickle cell disease, we would talk about the beta globin gene. The mutations are in the beta globin gene. And in our discussion, we would talk about normal alleles for beta globin, and we would also talk about mutant or sickle alleles for beta globin. So those two different variations in the DNA sequence that codes for the beta globin protein, those two variations would be referred to as two alleles of the beta globin gene. We will use the term allele a little later when we talk about polymorphisms in DNA markers. These are DNA markers are DNA sequences that don't necessarily do anything. They don't necessarily code for any protein, but they can be useful, for example, in genetic testing. And sometimes we will find such a DNA marker in which one DNA sequence might give a big band on a southern blot and a different variation of that same piece of DNA will give a small band on a southern blot. Those two variations would be referred to as two different alleles of that DNA marker. So we use the term allele when there are two or more variations of the same gene. The term genotype refers to the particular alleles that a person has at some locus. For example, again, if we were discussing sickle cell disease, in our discussion we would talk about people who are homozygous normal, having two normal alleles for beta globin. We would talk about people who are heterozygous or carriers. They would have, those people would have both a normal and a mutant allele for beta globin. And we would talk about people who are homozygous mutant or affected by that disease, having two mutant alleles for beta globin. So genotype refers to the particular alleles a person has for some gene. Now that genotype gives rise to a phenotype. The phenotype is the physically observable features. For example, are you male or female, or tall or short, or brunette or blonde, blue eyes, brown eyes, or for our purposes in medical genetics, are you normal, or do you have the symptoms of some genetic disease? And so that's how we will mostly use the term phenotype in medical genetics, normal as opposed to affected by the symptoms of some genetic disease. Homozygote or homozygous refers to a situation in which a person has two alleles that are the same for some gene. Homozygous normal, meaning having two normal alleles. Homozygous mutant, having two mutant alleles for some gene. Because we have pairs of chromosomes, we have two copies of almost all of our genes. Homozygote can be compared with the term heterozygote or heterozygous, in which the person has two different alleles for some gene. So the carriers for genetic diseases are heterozygous, having one normal and one mutant allele for a particular gene. The term dominant ordinarily means that we only need one copy of that particular allele in order to have the traits of that particular gene. We normally use the term dominant in medical genetics to refer to dominant genetic diseases. Ordinarily, for a dominant genetic disease, you only need to inherit one mutant allele in order to have the symptoms of the disease. That is compared with the term recessive. Ordinarily, the term recessive means that you need to inherit two mutant copies of that gene in order to have the symptoms of the disease. Some of the most famous genetic diseases, such as cystic fibrosis, sickle cell disease, Tay-Sachs disease, are examples of recessive diseases. The exception to the recessive, to requiring two copies of a recessive mutation to have a disease, is in X-linked recessive diseases in males. A male can inherit an X-linked recessive disease, have the symptoms of the disease, even though he has only one copy of the mutation, and that's because a male only has one X chromosome. And so if a male inherits a mutant X chromosome, he does not have a good X chromosome to hide the effects of that mutation. Let's also look at some of the basic kinds of mutations that will be causing all these genetic diseases that we'll be talking about. Missense mutations. Missense mutations are mutations or changes in our DNA sequence that lead to a single amino acid replacement in a protein. Sickle cell disease, for example, is the best, most famous example of a disease caused by a missense mutation. In the sickle cell disease, the mutation is in the gene coding for the protein beta globin. There is a mutation in the mutant beta globin allele that will change a glutamate to a valine in the beta globin protein. That's referred to as a missense mutation. Another well-known kind of mutation causing genetic diseases is a nonsense mutation. In a nonsense mutation, you remember that we have a DNA change that changes a codon for an amino acid to a stop codon so that the protein is too short and non-functional. We can also have deletions in which something is missing. We can have insertions in which we have extra bases. And if we have a single base deletion or insertion, we have a kind of mutation called a frame shift mutation. And you remember that what happens in a frame shift mutation is that as the ribosome is coming along trying to translate the messenger RNA, everything is okay until it comes to the point of that single base deletion or insertion, and at that point then the ribosome gets misaligned on the codons and cannot read the codons properly after the point of the mutation. So with a frame shift mutation, the first half of the protein is correct, the second half of the protein is just garbage, just a completely mixed up amino acid sequence, and leading to non-functional proteins. Now, also on your screen is the term in-frame. In-frame deletions or insertions are ones in which there is a deletion or insertion of some multiple of 3 bases. For example, the most common mutation that causes cystic fibrosis is a 3-base deletion. 3 bases are missing. With a 3-base deletion, essentially exactly 1 codon is missing from the gene, so that when the ribosome translates that messenger RNA molecule, it simply leaves out 1 amino acid, but all of the other codons can be read correctly, so that both before and after the mutation, the codons are read correctly. We call that an in-frame deletion or insertion. We refer to some of these kinds of mutations as loss of function mutations if they lead to a deficiency of something. Enzyme deficiencies, something is missing from the cell. It's referred to as a loss of function mutation. We also can have certain kinds of mutations that are called gain of function mutations. A gain of function mutation is one that leads to something extra being present in the cell. We can have a mutation that leads to the production of a completely new enzyme or an overproduction of a normal enzyme. We will meet gain of function mutations when we talk about oncogenes and their relationship to cancer. One of the most important things in Chapter 1 is analysis of pedigrees. Pedigrees are basically family histories that show us how particular genetic diseases are inherited within individual families. This slide shows the basic symbols that are used in pedigrees. You will see on the left side of your screen squares that represent males, circles representing females. If we don't know the sex of the individual, we usually use a diamond shape to represent that individual. Most commonly, the diamond shapes are used to represent fetuses. For example, in your exam, you will often see a pedigree or a description of a family in which the woman is pregnant, but we do not yet know the sex of the fetus. We ordinarily would represent that fetus then with a diamond shape. And often that diamond shape, that fetus, will be what the test question will be about. They will be asking you to make a prediction about the fetus. If the symbol is completely open, that means that the individual does not have the symptoms of the genetic disease, that is, they are normal. If the symbol is completely filled in, that person does have the symptoms of the disease, they are affected by the disease. Sometimes we indicate the carriers for genetic diseases, but this is optional and you will not always see this on your test questions. Carriers for autosomal recessive diseases are indicated by half filling in the symbol. The autosomal diseases are those carried on chromosomes 1 through 22, and normal individuals have 2 copies of each of those chromosomes. So carriers heterozygous carriers for those diseases might be represented by half filling in the symbol. Carriers for X-linked recessive diseases are always female and are represented by putting a dot in the middle of the circle representing that person. We will not go over all of the symbols in this particular figure, but on the right-hand side of the screen, I'd like to call your attention to the representation for an ordinary mating. We simply draw one single line between the mating couple. If there is consanguinity, or for example, if the couple is related to each other, if they're first cousins or something like that, then we will often draw two lines between the symbols for the mating couple. What will you be expected to do on the exam with these pedigrees that we'll look at? This is what I think the major test questions, the major topics for the test questions will be. You will be required to recognize the mode of inheritance. That is, on your exam, they will show you a pedigree and they will ask you to identify the mode of inheritance. That is the disease inherited in an autosomal dominant fashion, an X-linked recessive fashion, or what is the mode of inheritance? How will you determine that? Here's my simple-minded way. The first thing that I try to determine is whether a disease is dominant or recessive. What you look for in that case is the disease present in every generation, or does it skip generations? If the disease is found, if an individual with the symptoms of the disease is found in every generation, then the disease will be dominant. If the disease skips generations, then it will be recessive. The second thing you have to determine is whether the disease is autosomal or X-linked. Autosomal means that the gene is located on one of the autosomes, one of chromosomes 1 through 22. X-linked means it's carried on the X chromosome. What you look for to make that determination is male-to-male inheritance of the mutation. That is, does a father pass the mutation along to a son? If, in the pedigree, if you do find male-to-male, father-to-son inheritance of the mutation, then that disease will be autosomal. If you do not find male-to-male inheritance, then the disease will be X-linked. That's because males only have one X chromosome, which they must give to all their daughters. So a father, cannot pass an X-linked disease along to his sons. He can only give that mutation to his daughters. The kinds of modes of inheritance we've been talking about so far are all involved with diseases in which the mutations are found on our chromosomes found in our nucleus. Humans have about 30,000 genes, and the vast majority of those genes are found on the chromosomes in our nucleus. However, we also have a small amount of DNA in our mitochondria. The mitochondrial DNA does not carry very many genes, probably only about 40 or 50, but we can have mutations in those mitochondrial genes, and we can have diseases arising from those mitochondrial mutations. Mitochondria are inherited in a much different way from our chromosomes in our nucleus, leading to a different pattern of inheritance, which we will see in a few minutes. Mitochondrial diseases are inherited in a maternal fashion, that is, they are only passed through the mother. A second major thing that you will be asked to do on your exam with pedigrees is to predict the recurrence risk. That means on your exam they will show you a pedigree, or in words will describe a family, and they will say that the mother, the wife, is pregnant, and they will ask you to make a statistical prediction about the child. Is the child destined to have a disease, or to be a carrier, or to be normal? What is the probability that that child will be affected by this particular disease? That is called predicting the recurrence risk. We'll look at how to do that for each type of disease. The third thing that you may be asked to do on the exam is to identify obligate carriers. That is, you will be asked to identify those people in a family that must be a carrier for some genetic disease. Here's the first pedigree in your book, and if you look through the pedigree, you will see that there are affected individuals in every generation, so that will make the disease dominant. That will make the disease dominant. And if you look through the pedigree, you will also see inheritance from a father to a son. You will find that twice in this pedigree. Inheritance of the mutation from male to male, father to son inheritance. So that makes this a typical pattern for an autosomal dominant disease. Examples of autosomal dominant diseases are neurofibromatosis type 1, Huntington disease, Marfan syndrome. The couple that I've put the box around on your slide is a typical couple that you will find for an autosomal dominant disease. In that couple, one of the members, in this case it's the wife, is normal, that is, she is homozygous normal, does not have a copy of the mutation. The other member of the couple, in this case the father, is affected by the disease, and he is heterozygous for that disease. You can tell that he is heterozygous by looking back at his parents. He must have inherited the copy of the mutation from his father and a normal allele from his mother. Now, one of the typical kinds of questions that you might get on your exam would be, given such a couple, if the woman becomes pregnant again, what is the probability that their child will be affected by the disease? What is the, that they are asking you to make a prediction of the recurrence risk. The answer is 50%. The child would have a 50% chance of inheriting the mutation and of having the disease. Here is how we officially figure that out, by using something called a Punnett square. To draw a Punnett square, on the top we put the father's two alleles, and that represents essentially the two types of sperm that the father can produce. On the left we put the mother's two alleles. This represents the two kinds of eggs that she can produce. And then we simply fill in the four possible boxes with the four possible genotypes of the children that can be produced. And we color in the boxes of those children that would be affected by the genetic disease. There is something a little tricky about the Punnett squares that you should be aware of, and that is that we always represent the dominant allele with a capital letter, and we represent the recessive allele with a small letter. Since we're currently talking about an autosomal dominant disease, in this particular Punnett square, the mutation is represented by the capital A, and the normal allele is represented by the small a. I'm warning you about this because later on, when we talk about recessive diseases, the mutation will be represented by the lowercase letter and the normal allele will be represented by the capital letter. So don't get confused by that as we go through the various Punnett squares in this section. In this particular case, you can see that 2 out of the 4 boxes are colored in, so the recurrence risk in the child, the probability that the child would be affected by the disease, is 2 out of 4, or 1/2, or 50%. The recurrence risk is 50% for an autosomal dominant disease. Here's the next example in your book, and you can see that this disease has skipped many generations, so this is a typical pattern for a recessive disease. We also need to determine if there is male-to-male inheritance in this kind of pattern. This is a little more difficult to see. This particular pattern is for an autosomal recessive disease, and For an autosomal recessive disease, a person must inherit a copy of the mutation from the mother as well as a copy of the mutation from the father in order to have the symptoms of such a disease, so that the affected male child at the bottom right of your screen must have inherited a copy of the mutation from his mother and a copy of the mutation from his father. So since we do have father to son, male to male inheritance of the mutation, this would be an example of an autosomal recessive disease. Another way to identify this as autosomal recessive as opposed to X-linked recessive is the fact that we find in this pedigree both an affected male and female child. You will see later that for X-linked recessive diseases, it is almost always just males that are affected. You also will see something in this pedigree that we often, but not always, find in pedigrees for autosomal recessive diseases, and that is consanguinity. That is, the parents near the bottom of the pedigree are first cousins of each other, represented by the two lines drawn between the two symbols for them. You do not always find consanguinity for autosomal recessive diseases, but you often will find it. The reason that you often find it for these diseases is because such two first cousins have genes in common. That is, they share 1/8 of their genes from their common ancestors. So that if there is any mutation up in their common ancestors, then those two first cousins, because they share 1/8 of their genes, are more likely to both have a copy of the same mutation compared with random matings in the population. So they are more likely to give rise to children with genetic diseases than for random matings in the population. Now, that couple that I've put the box around in this pedigree is the most common high-yield couple for your licensure exam. This is a couple in which both the mother and the father are carriers, heterozygous carriers for an autosomal recessive disease, such as cystic fibrosis, Tay-Sachs, sickle cell disease. And a very common question on the licensure exam is, given such a family, if such a couple, if they have a child, what is the probability that their child will be affected by the disease? And we would officially figure that out by drawing a Punnett square. Again, the father's two alleles are at the top of this Punnett square. The mother's two alleles are on the side. Remember, since we are currently talking about an autosomal recessive disease, the mutation is represented by the lowercase a in this example. The normal allele is represented by the capital A. When we fill in all the boxes, an individual has to inherit two mutant copies, two mutant alleles in order to have the symptoms of the disease. And so the box at the lower right is colored in, and we would say that the probability of the child inheriting the disease is 1 out of 4, or 25%. We would also say that the child has a 50% chance of being a carrier, a heterozygous carrier for the disease, because 2 of the boxes would be carriers, and the child has a 25% or 1 in 4 chance of being homozygous normal. There is a variation of this kind of question that sometimes appears on the exam that many people have difficulty with, so I want to be sure that you know how to deal with that. On your exam, sometimes they will describe a family in which the father and mother are both carriers for an autosomal recessive disease such as cystic fibrosis, and the test question will tell you that this couple has had a child. You are the doctor, and you have a chance to go examine that child. As a result of your examination, you determine that the child does not have cystic fibrosis. That is, the child is normal, the child does not have the symptoms of the disease. The test question then will be, what is the probability that the child will be a carrier for this disease? The answer to that question is 2/3 or 67%. The reason for that is if you have had a chance to examine the child and you have determined that the child does not have the symptoms of the disease, then you have ruled out that colored square in the lower right portion of the Punnett square. That is, the child then must fit into one of the 3 white boxes in that diagram, since two out of the 3 white boxes represent carriers. If the child does not have the symptoms of the disease, the child will have a 2/3 probability of being a carrier for that disease. The next couple of examples are X-linked diseases. And when we talk about X-linked diseases, we always begin that topic by talking about something that happens with the X chromosome called X inactivation. X inactivation is something that occurs in all females. X inactivation is a mechanism that is used to equalize the amount of protein encoded by the X chromosomes in males and females. The basic idea here is that for all of the autosomes, all individuals, both males and females, have 2 copies of all of the autosomes. One copy that came from the father and the other that comes from the mother. And for the vast majority of our genes on our autosomes, both of those copies are active. That is, we express the genes and we make protein from both copies of those genes. Now, the problem is that females have 2 X chromosomes, but males have only 1 X chromosome. If all the genes on the X chromosomes were active in females, that would mean that the cells in a female would make double the amount of all those proteins encoded on the X chromosome compared with the cells in a male. But cells in a female require approximately the same amount of most proteins as males. So in order to prevent herself from making double the correct amount of these proteins, what happens early in the development of a female embryo is that randomly one or the other, that is either the paternal or maternal X chromosome, is methylated and inactivated, condensed to form a bar body, and turned off. The X chromosomes that are condensed to form bar bodies are not 100% completely inactivated. There are still a few genes that remain active, but the vast majority of genes on that X chromosome are turned off. Once that random inactivation occurs in a particular cell, that inactivation is fixed in all the descendants of that cell. That is, as that cell divides, it maintains the same pattern of inactivation that occurred early in development. So that in normal females, scattered throughout their bodies, roughly in half of the cells, the X chromosome that came from the father is turned off, and in the other half of her cells, the X chromosome that came from the mother is turned off. The last item on this slide says that all X chromosomes in a cell are inactivated except one. There are certain females that have more than 2 X chromosomes. For example, there are individuals that have 3 X chromosomes. Such an individual would have 2 bar bodies within each cell. Within each of her cells, randomly 2 of the X chromosomes would have become inactivated. This slide shows you a diagram just to illustrate this X inactivation. The X inactivation occurs very early in development of a female embryo at the blastocyst stage. That is when the early embryo consists of about 100 cells. And at the top of the diagram, what is indicated is that randomly within each cell, either the paternal or the maternal X chromosome is condensed to a bar body and inactivated and turned off. The graph at the bottom of the slide shows you that in a large population, the vast majority of females would have approximately a 50-50 distribution of such inactivation within their bodies. That is, within the body, in roughly half of the cells, the paternal X chromosome would be inactivated, and in 50% of the cells, the maternal X chromosome would be inactivated. However, since this inactivation occurs simply due to random chance, that is, the choice within an individual cell, the choice of which chromosome to inactivate is randomly chosen. There will be some individuals in which only a small number of cells have the paternal X chromosome inactivated, and there will also, at the other end of the scale, will be certain females that have only a small number of cells in which the maternal X chromosome was inactivated. That will occasionally give rise to an interesting phenomenon in which a female who is a carrier for an X-linked genetic disease can actually have the symptoms of the disease if, due to random chance, she was so unlucky that in her early development it was the vast majority of her normal X chromosomes that became inactivated and condensed to form Barr bodies. That is a pretty rare event, but it sometimes does occur. Here's a pedigree for one of these X-linked diseases, and in this pedigree we can see that the disease has skipped generations. That is, the oldest generation at the top of the screen does not have anyone with the disease, and on the left side of the screen we skipped the second generation from the bottom. Since we skipped generations, this would be a typical pattern for a recessive disease. If you look through this pedigree, you will not be able to find any examples of male-to-male, father-to-son inheritance of the mutation, and so that will make this an X-linked disease. So this is a typical pattern for an X-linked recessive disease such as hemophilia or Lesch-Nyhan syndrome or Duchenne muscular dystrophy, for example. The thing that really gives this pattern away as being X-linked recessive, however, is the fact that only males are affected by the disease. If you look through the pedigree, you will see 5 affected males, no affected females. The reason that we find almost only males being affected with X-linked recessive diseases is the fact that males have only one X chromosome. So that if a male inherits a mutant X chromosome, he will have the symptoms of that disease because he does not have a good X chromosome to hide the effects of the mutation. However, if a female inherits a mutant X chromosome, almost always she will have a normal X chromosome to hide the effects of that mutation. So for the typical pattern for X-linked recessive will show only males being affected. Another kind of question that they sometimes ask, particularly with regard to X-linked recessive pedigrees, is will often ask you to identify obligate carriers for the disease. An obligate carrier is someone who must be a carrier for this particular genetic disease. In this particular pattern, the mothers of affected sons would be obligate carriers. So, for example, at the very top of the screen, the mother, the female at the top of the screen, is an obligate carrier because she gave birth to affected sons. The other kind of obligate carrier for this pattern are daughters of affected males. On the right-hand side of the pedigree, at the bottom, you will see a daughter of an affected father. That daughter of an affected father is an obligate carrier because affected fathers must give their mutant X chromosome to all of their daughters, so all the daughters will be obligate carriers. On the left side of your screen, the daughters at the bottom over there are not obligate carriers, because their mother is an obligate carrier, but she has a 50% chance of passing along the mutant and a 50% chance of passing along the normal X chromosome to each daughter. So on the left side of your screen at the bottom, those daughters may be carriers. They have a 50% chance of being carriers, but they are not obligate carriers. The two couples that I've put boxes around in this pedigree are the typical couples that you may find on your exam for X-linked recessive diseases. On the left of your screen, the couple labeled B is the one that I think is the highest yield of all for this type of disease. This is a couple in which the father is normal and the mother is a carrier for the X-linked disease. In couple A, on the right-hand side of your screen, is a couple in which the mother is homozygous normal and the father is affected by the disease. You may see test questions that ask you to make predictions about the children of such couples, and the Punnett squares that make those predictions are shown on this screen. The highest yield one, in my opinion, is Punnett square B on the right side of your screen at the top. This was the situation in which the father was normal and the mother is a heterozygous carrier. In this particular example, since we're talking about an X-linked recessive disease, the small x, the lowercase x, represents the mutation in this case. And you can see that for such a couple, half of the sons would be affected by the disease. The son in the lower right part of the Punnett square, the box is colored in, so half of the sons would be affected by the disease. And on the left of that Punnett square, half of the daughters would be carriers for this disease. For the X-linked diseases, you should be very careful to determine if the test question asks you about sons or daughters or children, because the answers are different in all of those cases. If they ask about sons, you only count the two boxes on the right. You only count the male children. If they ask about daughters, you only count the two boxes that represent daughters. On the left side of your screen then is the Punnett square representing the results from a mating with couple A. Couple A was the one in which the father is affected by this disease, the mother is homozygous normal, and by looking at that Punnett square, you should be able to determine that all of the sons of such a couple will be completely normal. All of the daughters will be carriers, heterozygous carriers for the disease. All of the daughters are carriers because the affected father must give his mutant X chromosome to all of his daughters. The next pattern on your screen is the typical pattern for an X-linked dominant disease. If you look through this pedigree, you will see affected individuals in every generation, suggesting a dominant disease. You will not be able to find any male-to-male inheritance, identifying this as an X-linked disease. There are not very many X-linked dominant diseases. Fragile X syndrome is one well-known example. I put boxes around the two most common couples that you would find for an X-linked dominant disease. Couple A on the left of your screen is one in which the father is affected by the disease, the mother is homozygous normal. Couple B on the right side of your screen is one in which the father is normal and the mother is affected by the disease. Almost always for a dominant disease, when the mother is affected, she will be heterozygous, and you can determine that for the mother in couple B by looking back at her parents. She must have inherited the mutation from her mother and a normal allele from her father. So these are the high-yield, most common couples involved in X-linked dominant diseases. If you're asked to predict the recurrence risk in the children, these are the Punnett squares that would apply in this situation. For couple A, the Punnett square is on the left top of this screen. Remember, in couple A, the father was affected by the disease. The mother was homozygous normal. Remember, since we're talking about an X-linked dominant disease, the capital X represents the mutation. The normal allele is represented by a small x, so that for couple A, with a father being affected, all of his daughters will be affected by this disease because he must give his mutant X chromosome to all of his daughters, and since it is dominant, all the daughters will have the symptoms of the disease. All of the sons from that couple will be completely normal. On the right-hand side of the screen is the Punnett square representing the results from a mating in couple B. In couple B, the father was normal and the mother is affected by the disease but heterozygous. And I think you can see by looking at the boxes in the Punnett square that for that couple, half of all children would be predicted to be affected by the disease. It doesn't matter whether they are male or female. Half of all children would be affected by the disease. Now, the patterns we've been looking at, so far, the determination of the pattern of inheritance can be made by looking at this decision tree. This decision tree basically describes what we've already been doing as we've analyzed these pedigrees. If you look through this decision tree, what it will ask you at the top is the disease dominant or recessive? And if the disease is recessive, one of the way we identified then whether the disease is X-linked or autosomal is by looking for male-to-male, father-to-son inheritance. However, for the recessive diseases, it's actually easier to look at the children. If essentially only males are affected for a recessive disease, then the pattern will be X-linked recessive. If there are both affected males and females, then the pattern will be autosomal recessive. For the dominant diseases, we would look for male-to-male transmission of the mutation, and if we do find male-to-male transmission, the disease will be autosomal. If we do not find male-to-male inheritance of the disease will be X-linked. These are the patterns that we have for diseases carried on our chromosomes found in our nucleus. However, there is another kind of disease called mitochondrial diseases, which show a completely different kind of inheritance. These are diseases in which the mutations are found in our mitochondrial DNA. Our mitochondria have small circular DNA molecules that carry a relatively small number of genes, perhaps 40 or 50 genes in the mitochondrial DNA. However, there can be mutations in that mitochondrial DNA that can lead to genetic diseases. Our mitochondria provide energy for our cells. That is, they make most of the ATP used by our cells. So if we have a mitochondrion with a mutation, those mitochondria tend not to work very well and they do not produce enough energy. So the mitochondrial diseases tend to show their effects, that is, the symptoms show up in those organs or those tissues that require the most energy. And that comes down mostly to muscles and nerves. Muscles and nerves require more energy than most other tissues. So examples of mitochondrial diseases such as Leber hereditary optic neuropathy will show up in this case, in nerves, the optic nerve. Leber hereditary optic neuropathy is a disease in which the optic nerve deteriorates because it's not getting enough energy from the mitochondria, and the individuals go blind when they become about 20 or 25 years old. Now, the inheritance pattern for mitochondrial diseases is much different than it is for the diseases carried on our chromosomes, because we get our mitochondria only from our mothers. Fathers do not contribute any mitochondria to the children. And so, in this pattern on your screen, you can see that if we have an affected mother, such as the mother at the very top of the screen, if we have an affected mother, she will give her mutant mitochondria to all of her children, so all of her children will be affected. And in the middle and lower right parts of the screen, you can also see each of those affected mothers has only affected children. On the left part of your screen, you will see an affected father, and I think you can see that none of his children will be affected, because mitochondrial diseases show maternal inheritance. They are only inherited through the mother. So far we've talked about pedigree analysis, and remember, as you study these pedigrees, focus on the material that you expect to see on the exam. On your exam, you will be asked to identify the mode of inheritance shown by particular pedigrees. You will be asked to identify obligate carriers in the pedigree, and you will be asked to predict the recurrence risk for these diseases. After looking at all of those pedigrees in the beginning part of Chapter 1, in your book you will come to a section entitled Important Principles That Can Characterize Single Gene Diseases. Now, if I had personally written this chapter, I would have entitled this section Things That Make It a Whole Lot More Complicated and Mess You Up, because these things that we're going to look at in the rest of this chapter are complications or things that don't fit those basic patterns that we talked about earlier. They're things that make it more complicated and make it harder to analyze your pedigrees. But I want to be sure that you understand what to do on your exam. On your exam, if they simply show you a pedigree and ask, what is the mode of inheritance, then you should forget about all these complications and just simply apply those basic rules that we looked at earlier. On your exam, if they want to ask you a question about one of these complications, they will call your attention to it in some way. So we'll go through these complications one at a time. There's a long list of them: variable expression, incomplete penetrance, pleiotropy, locus heterogeneity, new mutation, delayed age of onset, anticipation, and imprinting. We'll look at them one at a time. Let's start with variable expression. The term variable expression just means that not everybody who has a particular genetic disease will have the same severity of symptoms. That is, some individuals will have very severe symptoms, others might have much milder symptoms of the disease. Now, sometimes that difference in severity of symptoms can be as a result of the particular kind of mutation that an individual has. For example, if an individual has a genetic disease that resulted from missense mutations, sometimes those symptoms will be milder than someone who has their particular disease resulting from nonsense or frameshift mutations. And that's because with missense mutations, since they result in just amino acid replacements, sometimes the particular enzyme involved may still be partially active, leading to milder symptoms of the disease. However, if an individual has a disease resulting from nonsense or frameshift mutations, those kinds of mutations almost always lead to complete enzyme deficiencies and more severe symptoms. Some examples of diseases showing variable expression are hereditary hemochromatosis and neurofibromatosis type 1. Hereditary hemochromatosis is a disease, an inherited genetic disease, in which the patients accumulate too much iron in their bodies. And this iron can form deposits all over the body. the particular symptoms that an individual has will depend on how much iron is deposited and where in the body it is deposited. So these patients will often have symptoms of arthritis due to iron deposits in their joints. They'll have diabetes due to iron deposits in the pancreas. They'll have heart disease because of iron deposits in their heart, for example. And we refer to this as an example of variable expression because the severity of symptoms will depend on how much iron gets deposited during the lifetime of that individual. The amount of iron that gets deposited is dependent on several factors. One factor is how much iron is taken in during your lifetime, and that would depend on how the food you eat, the water you drink, and whether or not you take iron supplements. The other important factor is how much iron you get rid of during your lifetime, and that is dependent mostly on whether you are male or female. Females get rid of a lot of iron during their lifetimes by menstruation, pregnancy, and lactation. On the other hand, you might have a male who's a really good citizen and goes down to the Red Cross and donates blood every 56 days, the way they would like for you to. And by making all those blood donations, he would be accidentally protecting himself from hemochromatosis by getting rid of a lot of iron during his lifetime. So the severity of symptoms, the appearance of the disease, can depend on your sex as well as the amount of iron you've taken in during your lifetime. Another example of variable expression is neurofibromatosis type 1. This is a picture of an individual with this disease. Neurofibromatosis type 1 is a disease in which patients develop little benign tumors at the nerve endings. If you look at this person, you will see a lot of little bumps or tumors scattered all over his body. Each of those little tumors is a benign tumor at a nerve ending. We refer to this, or we say that this disease has variable expression because some patients have such a small number of these bumps of such a small size that they do not even know that they have the disease. At the other extreme, I recently read a newspaper article about a woman with this disease in Europe who had one of the tumors removed, and the tumor weighed 156 pounds. What was left of her weighed 76 pounds. So obviously there's an extreme amount of variable expression in the severity of the symptoms for this disease. When we use the term variable expression, everyone with the disease has symptoms of some kind, but the severity differs from person to person. The second complication is incomplete penetrance. In incomplete penetrance, we can identify some individuals who have the disease genotype but do not display the disease phenotype. What that means is sometimes for certain diseases you can analyze a pedigree, and by analyzing that pedigree you can point to a particular individual and say that person has the mutation. That person should have the disease, but they are perfectly fine. They have no symptoms whatsoever. That is referred to as incomplete penetrance, when the person has inherited the mutations, has the genotype that says they should have the disease, but do not. We will use the term degree of penetrance when we discuss this kind of disease. The degree of penetrance essentially means the percentage of people who inherit the mutation who also go on to develop the disease. In just a few minutes, I'm going to tell you about a disease type of disease called retinoblastoma, and we will say that the inherited form of retinoblastoma shows 90% penetrance. That means that if you inherit the mutation, you have a 90% chance of developing the disease, but you also have a 10% chance of getting lucky and not showing any symptoms at all. The diseases showing incomplete penetrance are usually those in which some sort of secondary event or secondary influence is required in addition to the inheritance of the mutation to cause expression of the disease. Here is a pedigree found in your book for incomplete penetrance. And the example I like to use for this pedigree is retinoblastoma, cancer of the developing retina. The inherited form of retinoblastoma is caused by mutations in a tumor suppressor gene called the RB gene. Tumor suppressor gene called the RB gene. Now, the inherited form of retinoblastoma is inherited in an autosomal dominant fashion. And that's a fact. But if you look at this pedigree, you will notice something unusual in this pedigree in that the male that the arrow is pointing to has been skipped. That is, this is an autosomal dominant disease. We know that that male has inherited a copy of the mutation because he has passed it along to his two sons. So he has a copy of the mutation. It's autosomal dominant. We would predict that he should have the symptoms of the disease, but he does not. He's perfectly fine. That is an example of incomplete penetrance. That male simply got lucky and did not develop the symptoms of the disease. How can we explain something like that? Retinoblastoma is caused by mutations in the RB tumor suppressor gene. Now, one of the basic principles of tumor suppressor genes and their relationship to cancer is that in order for mutations in a tumor suppressor gene to lead to the development of cancer, within an individual cell, we must mutate or knock out both copies of that same tumor suppressor gene within the same cell. Remember, since we have pairs of chromosomes, we all have 2 copies of most of our genes. So normal individuals have 2 copies of the RB gene. What is being inherited in this family is 1 mutant copy of the RB gene, so that each of the people in this pedigree who is affected by the disease, as well as that male identified by the arrow who does not have the disease, each of those people is heterozygous for the RB locus. That is, each of those people has 1 normal and 1 mutant RB allele. In order for the disease retinoblastoma to develop, what must occur during the early development of the retina is that at least 1 single cell in that developing retina must acquire a secondary mutation, a secondary somatic cell mutation that knocks out or mutates the other copy of the RB gene within that 1 single cell. And so if that happens, and it might happen as a result of exposure to radiation or chemicals or just simply bad luck during DNA replication, if that single cell does acquire that secondary mutation, then we say that that 1 cell has lost its heterozygosity. That is, that 1 single cell is not heterozygous anymore. That 1 single cell has 2 copies of the mutant RB allele, and that 1 single cell then will multiply to form the tumor that we identify as retinoblastoma. And it turns out that for all those individuals in such a family that inherit a copy of the mutation, if you inherit 1 copy of the mutation, you have a 90% probability that within 1 single cell you will acquire that secondary mutation that will knock out the other copy of the RB gene within that cell and will lead that cell to become a tumor. So that we say that retinoblastoma is 90% penetrant. That is, 90% of the people that inherit the first mutation will go on to develop the symptoms of the disease. We say that this disease is autosomal dominant because the individuals are only inheriting one copy of the mutation. The second copy of the mutation is acquired, an acquired somatic cell mutation. There are some other inherited forms of cancer that follow exactly the same kind of pattern. For example, the inherited forms of breast cancer involving the tumor suppressor genes BRCA1 and BRCA2 follow exactly this same kind of pattern. The inherited forms of breast cancer are about 70% penetrant, so that if a woman inherits one mutant copy of BRCA1 or BRCA2, she will have about a 70% chance of acquiring that secondary mutation sometime during her lifetime and developing breast cancer. Another example of such an inherited syndrome is Li-Fraumeni syndrome, involving inheritance of a p53 tumor suppressor gene. We will discuss that one later on in Chapter 4 when we talk about multifactorial diseases. Another complication is pleiotropy. Pleiotropy refers to the fact that one single disease-causing mutation can affect many different organs at once. Pleiotropy is very common among genetic diseases. In fact, pleiotropy probably occurs in essentially all genetic diseases. A good example of pleiotropy is Marfan syndrome. Here's a picture of a kid with Marfan syndrome. Marfan syndrome is an autosomal dominant disease. It occurs in about 1 in every 10,000 people. It's caused by mutations in a gene that codes for a protein called fibrillin. Fibrillin is a structural protein, and that structural protein, fibrillin, is found throughout the body. So the idea with pleiotropy here is that anywhere in the body that fibrillin is located, the symptoms for Marfan syndrome can appear. And you can see some of the characteristics of these patients just by looking at the patient. These patients are very tall and thin. They have long, thin arms. And when they extend their fingers, they are said to have very long, spidery fingers. But there are other parts of their body that are affected also that you cannot see. These patients often, for example, will have aortic aneurysms because fibrillin is a component of blood vessels, and those weakened blood vessels can lead to aneurysms in these patients. We can also find examples of pleiotropy in almost any genetic disease. If you think, for example, about cystic fibrosis. In cystic fibrosis, we usually think of the symptoms of that disease appearing in the lungs, but we also know that the symptoms, problems with cystic fibrosis can occur in the pancreas as well, and essentially anywhere in the body that there's a mucous membrane that can get clogged up, such a patient will have problems. So pleiotropy means one single mutation, but can lead to problems in many different parts of the body. Another complication is locus heterogeneity. Locus heterogeneity is a situation in which we have one disease phenotype, or one disease that can be caused by mutations at 2 or more completely different genes. One disease caused by mutations at 2 or more completely different genes. Often these diseases involve genes that code for different subunits of a multi-chain protein. A good example is osteogenesis imperfecta, or brittle bone disease. On your exam, you may see this as a little clinical vignette that describes a child that has multiple broken bones in its first few months of life, maybe some skeletal deformities. And another thing to look for is blue sclera. That is, the whites of the eyes actually appear blue in this disease. Well, it turns out that this disease, osteogenesis imperfecta, brittle bone disease, is caused by mutations in collagen genes. But it turns out that some of these patients have the disease as a result of mutations in collagen genes found on chromosome number 7, whereas other patients with exactly the same symptoms have their disease as a result of mutations in collagen genes found on chromosome number 17. Two completely different genes on two completely different chromosomes, but mutations in those two completely different genes giving rise to the same disease. That is an example of locus heterogeneity. Another complication is new mutations. When we were analyzing those pedigrees at the beginning of Chapter 1, we assumed that all the mutations in those pedigrees were already existing and were simply being passed along from generation to generation. If a new mutation were to occur, however, that would completely mess up our ability to analyze a pedigree. I want to assure you, as you're taking your exam, that they will not do that to you on the exam. On the exam, if you are shown a pedigree and are asked to identify the mode of inheritance, you should assume that all the mutations are already existing and are just being inherited within that family. They will not be throwing a new mutation into the middle of that pedigree. It would be impossible to identify the mode of inheritance if they were to do that. Now, the one thing that you may be expected to know about new mutations refers to the recurrence risk for other members of the family. If a patient does have a genetic disease as a result of a new mutation, that child develops that disease simply as a result of an accident that happened probably in the egg or the sperm that led to that child. It was a random accident in that gene that is not likely to occur again, so that makes the recurrence risk to other siblings of that child very low. That is, if a child has a genetic disease as a result of a new mutation, it is not likely that those parents will have additional children with that same problem. However, once that child grows up to have children of his or her own, then that mutation can be passed along to the future generations, just like any other mutation. So the risk to siblings of the original child is very low, but risk to the future generations follows the normal patterns of inheritance that we looked at earlier. This diagram shows a pedigree in which a new mutation is occurring. All the people in the older generations have perfectly normal genes. The child, the son there on the male child in the lower right part of this diagram has a disease, perhaps Duchenne muscular dystrophy or something like that, as a result of a new mutation that occurred in the egg or sperm that led to that child, most likely in the egg. It would be impossible, just looking at the pedigree, it would be impossible to tell whether that child resulted from a new mutation or whether it was simply a recessive inherited disease. So they will, when the test writers make up the pedigrees, make up the test questions, they will not be showing you new mutations like this and expect you to analyze the pedigree. Another complication is delayed age of onset. This is a term that is used to refer to diseases in which the symptoms do not show up until the person gets older. There are certain diseases such as Huntington's disease, myotonic dystrophy, and some others in which the symptoms don't show up until the person is perhaps 40 or 50 years old. The fact that the symptoms don't show up until later in life just simply makes it difficult to analyze pedigrees and to give good genetic counseling to your patients. The idea here is that perhaps an individual might know that a disease such as Huntington's disease is being inherited in his family, and that patient might come to you for advice, but perhaps he might come to you advice when he's 20 years old, and he simply is not old enough yet to be showing the symptoms of this disease. It makes it very difficult for you to give your best genetic counseling to him because he simply is not old enough yet to be showing the symptoms of the disease, and so you do not yet know whether or not he has inherited the mutation. And another problem with this is that often the symptoms for these kinds of diseases, such as Huntington's disease, the symptoms do not show up until after the patient has already had children. So simply, delayed age of onset simply makes it difficult to give good genetic advice to patients, and it simply delays by many years your ability to completely analyze a pedigree. Huntington disease is a disease in which the patients are typically perfectly fine until they're about 40 years old or so, and then they begin to develop behavior problems and other neurological problems, and over the course of about 15 years or so, these patients just simply die a horrible death because of the complete deterioration of their neurological system. Diseases such as Huntington's disease and some of the other diseases with delayed age of onset are some of the ones that have benefited dramatically from our new ability to now do genetic testing or DNA testing for these diseases. Huntington's disease was one of the very first diseases in which a genetic test was developed. And this means that now with the genetic test, we can determine at any age whether a patient is destined to show Huntington's disease. And in fact, we can even test a fetus to determine whether that fetus is destined to develop this disease. So now a patient does not have to wait until he or she is 40 years old to determine whether or not they have inherited such a mutation, if they choose to know. But there are many people who will choose not to have such genetic testing because they simply do not want to know whether or not they have inherited such a mutation for such a terrible disease. Another complication is anticipation. Anticipation is something that we've known about for many years genetically, but we have only really been able to explain at the molecular level for about the past 15 years or so. Anticipation occurs with certain diseases such as myotonic dystrophy, Fragile X syndrome, and Huntington's disease. And it is a situation in which, if we look at pedigrees of families in which these diseases are inherited, what we see is, as we go down through the generations in such a family, the people affected with the disease get their symptoms at earlier and earlier ages as we come down to the most recent generations, and most commonly the symptoms become more and more severe as we come to the more recent generations. That phenomenon is known as anticipation. Essentially what is happening is that the mutation in these families is getting worse and worse and worse as it moves down through the generations in a family. And now we know that anticipation is caused by trinucleotide repeat expansions. What that means is that there are many places in our DNA where we have repeated sequences of DNA. That is, we will have the same DNA base sequence repeated several times in a row. If that sequence of DNA bases is 3 bases long, then we refer to that as a trinucleotide repeat. And sometimes, those are perfectly normal. We all have many, many trinucleotide repeats in our genomes. That's perfectly normal. However, sometimes, most likely during DNA replication, sometimes those trinucleotide repeats can cause a problem because it seems as if DNA polymerase gets confused when it's replicating the DNA for such a repeated region, and it appears to accidentally make too many copies of that trinucleotide repeat. So the idea is, we say that if that occurs and too many copies of the repeated region are made, we say that that trinucleotide repeat has expanded. And that expansion, that trinucleotide repeat or triplet repeat expansion, is the mutation. And once that expansion has occurred, that expansion tends to occur even more with each generation. That is, the mutation gets bigger and bigger and bigger. The mutation gets worse as we go down through the generations in a family. And as the mutation gets worse, the symptoms begin appearing at earlier and earlier ages in the more recent generations, and typically the symptoms become more severe in the most recent generations. And again, the most common examples of anticipation caused by trinucleotide repeat expansion are myotonic dystrophy, Fragile X syndrome, and Huntington's disease. Here is a pedigree, an example of a pedigree showing anticipation for Huntington's disease. Huntington's disease is autosomal dominant, and if you look through this pedigree, what you will see is for each of the affected individuals, the first number shown under those affected individuals is the age at which their symptoms first appeared. So the male nearest the top of the screen who first showed the disease in this family had his symptoms begin to appear at about age 65. His children, his affected children, had their symptoms appearing at around ages 53 or 55, and in the most recent generation then, symptoms began to appear at about age 40. So symptoms began to appear at earlier and earlier ages, and the symptoms most likely were more and more severe as we come down through those generations as well. The number in parenthesis after or underneath each affected individual shows the number of copies of the particular triplet repeat in these patients. The particular trinucleotide that is repeated in Huntington's disease is CAG. So for example, the male nearest the top had originally had 37 of these repeated regions. His children had 39 or 40 of those repeated copies, and the female at the bottom of this pedigree had 44. So this triplet trinucleotide repeat was expanding. The mutation was getting worse as it went down through the generations in this family. The last complication in this chapter is a complication called imprinting. It's also known as genetic imprinting or genomic imprinting. Genetic imprinting is a situation in which we have certain genes that are only active when they are inherited by one or the other, from one or the other parent. That is, we have certain genes that are only active when they are inherited from the father, and other genes that are only active inherited from the mother. Now, humans have about 30,000 genes, and we have 2 copies for the vast majority of those genes. One copy comes from the father, and the other copy comes from the mother. Now, for the vast majority of those 30,000 genes, both copies are active. That is, both the copy from the father and the copy from the mother are active. The genes are transcribed. They produce enzymes or protein molecules from those genes. However, we are beginning to discover a few genes in which that is not true. We're beginning to discover a few genes that we say are imprinted by either the father or the mother and turned off so that such a gene is not expressed. The copy of the gene from one parent is not expressed. Only the single copy from the other parent is expressed. Such imprinting occurs during gametogenesis, that is, it occurs as we are producing our eggs and sperm, and we have identified certain particular genes that are imprinted by males when they produce their sperm, and that gene then remains turned off for the life of the child that develops from that sperm. There are other different genes that are imprinted and turned off by a female as she is producing her eggs, and then that gene, that particular imprinted gene, will remain turned off for the complete life of the child that results from that egg. Such imprinting or inactivation is caused mostly by methylation of that gene. Methylation, imprinting, turning off the gene. Now, this is perfectly normal, and we all have, normal individuals all have examples of such imprinted genes. However, a problem can develop if a deletion or other mutation occurs in the chromosome that comes from the parent that is supposed to give us the active copy of the gene. If a deletion or mutation occurs in the chromosome from that parent that is supposed to give us the active copy, then the child will have no active copies of that gene and a disease will result. That is, the child will have no active copies because the gene from one parent is mutated or deleted. The gene from the other parent is methylated, imprinted, and turned off, so the child has no active copies and will show the symptoms of some disease. Another way that this will often show up on exams is examples of diseases in which we have one single deletion, one single chromosomal deletion that can give rise to two completely different diseases depending on whether that deletion is inherited from the father or the mother. The favorite examples for genetic imprinting are Prader-Willi syndrome and Angelman syndrome. Here's a picture of a child with Prader-Willi syndrome. Prader-Willi syndrome is a syndrome that is caused by inheritance of a deletion from chromosome number 15, and this syndrome results if that chromosomal deletion is inherited from the copy of chromosome 15 that comes from the father. These children are typically moderately mentally retarded. They are obese, hypotonic, show hypogonadism, small hands and feet. A child will have this disease if the deletion is on the copy of chromosome 15 from the father. Now, if that exact same deletion comes from the mother, then the child will have a completely different disease called Angelman syndrome. The Angelman syndrome children are typically very severely mentally retarded. They have a lot of neurological problems. You may notice in this picture the smile on this child's face. These children are usually very happy. They have a happy, pleasant disposition. You may also notice the way he's holding his hands and his arms. These children are often referred to as happy puppet children, or having the happy puppet disease, because of the way they hold their hands and arms, and because of their jerky movements. They look as if they are puppets, or puppets being jerked around on a string. So it's sometimes called the happy puppet disease. So, how can we explain this? How can we have two completely different diseases caused by inheritance of the same mutation, just depending on whether that mutation, that deletion, comes from the father or the mother? Here is the idea. This diagram indicates, is meant to indicate the inheritance of chromosome number 15, and this diagram is indicating that on chromosome number 15 there are two genes that are relatively close together. Those two genes are identified as, or we will call, them the Prader-Willi gene and the Angelman gene. In this particular picture, I've identified the Prader-Willi gene with the letters PW. It's represented by the rectangles in this picture. Nearby that Prader-Willi gene on chromosome 15 is a second completely different gene known as the Angelman gene, represented by the ovals in this picture. I've indicated the Angelman gene by the letter A in this diagram. Now, the idea is that the Prader-Willi gene and the Angelman gene are two genes that show genetic imprinting. It turns out that the Prader-Willi gene is methylated, imprinted, and turned off by the mother as she produces her eggs. And that is represented by filling in the rectangle for the Prader-Willi gene on the copy of the chromosome that comes from the mother. In this diagram, the copy of chromosome 15 coming from the father is on the left. The copy of chromosome 15 coming from the mother is on the right. And for the Prader-Willi gene, the open rectangle indicates that the gene coming from the father is active, working, produces a functional protein. The rectangle on the chromosome 15 coming from the mother is filled in, indicating that the mother has methylated, imprinted, and turned off that Prader-Willi gene in her chromosome. Now, that this child will be okay because the child does receive a normal functional Prader-Willi gene from the father. Also, a normal individual will inherit a copy of the Angelman gene from both the father and the mother. However, the Angelman gene is imprinted by males when they produce their sperm, and that is indicated by filling in the oval representing the Angelman gene on the copy of chromosome 15 coming from the father on the left. The Angelman gene on chromosome 15 from the mother is active and working, produces functional proteins, and so a normal individual will be okay, that is, will not have any symptoms, because, as this diagram indicates, a normal individual will inherit a normal functional Prader-Willi gene from the father and a normal functional Angelman gene from the mother. So such a situation is normal. That has occurred in all of us. We are okay because we do have at least one active copy of each of these genes. However, problems will occur if we have a deletion or mutation on the chromosome from the parent that is supposed to give us the functional copy of that particular gene. In this diagram, it indicates how such a deletion or mutation could lead to the production of Prader-Willi syndrome. What this diagram indicates is that the Prader-Willi gene has been deleted from the copy of chromosome 15 that is coming from the father. The Prader-Willi gene is still present on the copy of chromosome 15 from the mother, but the mother has methylated, imprinted, and turned off that Prader-Willi gene, so that in this situation the child would receive no functional copies of the Prader-Willi gene, and the child would develop Prader-Willi syndrome. The next diagram illustrates how Angelman syndrome could appear. What is indicated in this diagram is that the copy of chromosome 15 coming from the mother has a deletion that has removed the active normal Angelman gene. The copy of chromosome 15 coming from the father is still there. However, remember that males methylate, imprint, and turn off the Angelman gene as they are producing their sperm, and so the child does not inherit any functional copies of the Angelman gene. The Angelman gene was missing in the chromosome that comes from the mother. It's turned off in the chromosome that comes from the father. Since the child has no functional copies of the Angelman gene, the child will develop Angelman syndrome. The reason that we can get these two completely different diseases from one single deletion is that we can have one deletion that can remove both of those genes at once. Since the Prader-Willi gene and the Angelman gene are located close by each other, one single deletion can remove both of them at once. However, it's important to remember that they are two completely different diseases. And the idea is the reason we can get two completely different diseases from one deletion is that the Prader-Willi gene is imprinted in mothers as they are producing their eggs. The Angelman gene is imprinted by fathers as they are producing their sperm. So we have two different patterns of imprinting in the two completely different diseases. Now, on the exam, this is most likely to show up as a situation in which there is one mutation or one deletion that gives rise to two completely different diseases, depending on whether that mutation is inherited from the father or the mother. That would be an example of imprinting. And the other way to approach those test questions is if you see any test question that asks you about Prader-Willi syndrome or Angelman syndrome, you simply pick imprinting and go on to the next question. You will get it right, because almost always Prader-Willi and Angelman syndromes are the examples used for genetic imprinting. So, in this part of Chapter 1, we've reviewed some of the complications that can make it more difficult to interpret pedigrees, to analyze pedigrees. But again, I want to remind you of what to do with these complications. On your exam, if they simply show you a pedigree and ask to identify the mode of inheritance, then you should forget about these complications and just apply the basic rules that we applied in the first part of Chapter 1. On your exam, if they want to ask you about one of these complications like incomplete penetrance or imprinting or any of these complications, they will somehow call your attention to that complication so that you will know how to focus your attention on the test question. The next topic in our book is population genetics. I'm Dr. Vernon Reichenbaker from Marshall University, and we'll be talking about population genetics, also sometimes known as the study of genetic variation in populations. Now, in this topic, we will continue our discussion of single gene diseases. In Chapter 1, we were talking about single gene diseases and how they are inherited within individual families, analyzing pedigrees. In Chapter 2, in the topic of population genetics, we will still be looking at single gene diseases. However, here we will be looking at how those diseases behave in large populations, the kinds of calculations that the test writers will expect you to be able to do for your Step 1 exam. In this topic of population genetics, there are some terms that we need to learn. These two terms are genotype frequency and allele frequency. Genotype frequency refers to the proportion of each genotype in a population. What that means is this. Suppose we were talking about sickle cell disease. If we were talking about sickle cell disease in a population, in our discussion, we would talk about people who are homozygous normal. We would also talk about people who are heterozygous or carriers for sickle cell disease. And we would also talk about people who are homozygous mutant or affected by sickle cell disease. So, in our discussion of sickle cell disease, we would talk about people with three possible different genotypes. The term genotype frequency would refer to what fraction or what percentage of the whole population is found within each genotype? What fraction of a population is homozygous normal? What fraction of a population is affected by the disease? That's genotype frequency. Allele frequency refers to the proportion of chromosomes that contain a specific allele in a population. Another way of saying that is this. Of all of the copies of a particular gene in the world, what fraction of those genes are represented by each possible allele? So again, for our example with sickle cell disease, if we, in our discussion of sickle cell disease, we would be talking about two different possible alleles for the gene beta globin. We would talk about normal alleles for beta globin and sickle or mutant alleles for beta globin. So in our discussion, there would be two possible alleles. Allele frequency would mean of all those copies of the beta globin gene in the world, what fraction or what percentage of them are normal and what fraction or what percentage of them are mutant or sickle. So genotype frequency and allele frequency are terms that we will use throughout this chapter. Now, to illustrate what we mean by those terms, genotype frequency and allele frequency, in your book we discuss an example using restriction fragment length polymorphisms, or RFLPs. RFLPs are kinds of genes that we call DNA markers. They're genes that don't necessarily do anything, but they are genes that we can identify in the lab and detect by a technique called Southern blotting. And for the particular example shown here in this slide, this particular RFLP gene has two different alleles. One variation of its DNA sequence, when detected by Southern blotting, would give a larger DNA band, or an upper band, on the Southern blots indicated here. We will call that allele number 1. And the other variation of that DNA sequence for that marker will give us a lower or smaller molecular weight DNA band on a Southern blot, identified as allele number 2 in this slide. Now, since normal individuals have pairs of chromosomes, we each have 2 copies of most of our genes, including 2 copies of this particular RFLP marker. So that in the population, there will be people with 3 possible genotypes for this RFLP allele, or this RFLP gene. There will be people who are homozygous 1-1, people who are heterozygous 1-2, and people who are homozygous 2-2. The people with each of those 3 different genotypes will give different patterns of bands on the Southern blots, as indicated here. And so this is the way that we can identify which genotype a particular person has. In your book, in the example, what they have done is to take DNA samples from 100 people. Down at the bottom it says the total count is 100. That means that they took DNA samples from 100 people, ran those Southern blots to determine the genotype of each of those 100 people. And you can see in this table that they found 49 people with the genotype 1-1, 42 people with the genotype 1-2, that is the heterozygous people, and there were 9 people who were homozygous 2-2. To determine the genotype frequency for each of those 3 genotypes, what we do is divide the number of people with each genotype by the total population, which in this case is 100. And the right-hand column in this table shows the 3 genotype frequencies that are calculated from this basic data. For example, there were 49 people out of 100 with the genotype 1-1, so the genotype frequency for the homozygous 1-1 people is 0.49, or 49%. 49% of this population is homozygous 1-1. And we do a similar calculation to calculate the other genotype frequencies. Now that we have determined the genotype frequencies, we can also determine the allele frequencies in this example. And the calculation of allele frequencies is indicated on this next slide. The first two columns in this table are the same as we looked at in the previous slide. That is, we have a column that shows the 3 possible genotypes and the number of people within each genotype. Because we have pairs of chromosomes and 2 copies of most of our genes, in this population of 100 people, there would be 200 total copies of this particular RFLP gene. And in order to determine allele frequencies, what we have to do is determine what fraction of these 200 genes are represented by allele 1 and what fraction are represented by allele 2. The calculation of those allele frequencies is shown in the right-hand part of this table. Each of the homozygous 1-1 individuals will have 2 copies of allele number 1. Since there are 49 of those people, in those 49 1-1 people, there would be a total of 98 copies, 2 times 49, or 98 copies of allele number 1. Each of the heterozygous 1-2 people would have 1 copy of allele number 1. Since there are 42 of those people, that would be an additional 42 copies of allele number 1. So we add up all the copies of allele number 1 in this population and divide by 200 the total number of copies of this gene in the population, and when we grind through the math, we get 0.7 as the allele frequency for allele number 1. That means that of all the copies of this gene in this population, 70% of them, allele frequency 0.7, or 70% of them, of those genes, are allele number 1. We can do a similar calculation as shown near the bottom right of this table for allele number 2 to calculate that the allele frequency for allele number 2 is 0.3. That is 30% of the genes of this RFLP gene in this population are represented by allele number 2. We could have determined allele number 2 in a quicker way. That is, after we calculate allele number 1, we can get the allele frequency for allele number 2 just simply by subtracting from 1. So for allele number 2, we would have subtracted 0.7 from 1 to get our answer of 0.3. Now, the way this kind of information becomes really important to us in medical genetics is through something known as the Hardy-Weinberg equilibrium and the Hardy-Weinberg equation. The Hardy-Weinberg equation is listed here at the bottom of this slide. P squared plus 2pq plus q squared equals 1, and you will be expected to have memorized this equation. What Hardy and Weinberg determined is that if a population is large, and if we have random mating in that large population, then there should be a predictable relationship between those genotype frequencies and allele frequencies for all the for essentially all genes. Basically, this is saying there should be a mathematical way of predicting the numbers for genotype frequencies and allele frequencies. And that mathematical relationship is represented there by the Hardy-Weinberg equation, p squared plus 2pq plus q squared equals 1. This is an interpretation of that Hardy-Weinberg equation for the example using the RFLP that we were just talking about. In the Hardy-Weinberg equation, P and Q represent the 2 allele frequencies. So in our RFLP example, P would represent the allele frequency for allele number 1, Q represents the allele frequency for allele number 2. The three terms on the left side of that Hardy-Weinberg equation, p squared, 2pq, and q squared, represent the three genotype frequencies. P squared represents the genotype frequency for the homozygous 1,1 people. 2pq represents the genotype frequency for the heterozygous 1,2 people. Q squared represents the genotype frequency for the homozygous 2,2 people. Now, we had just determined a few minutes ago that the values for P and Q, that is the two allele frequencies in our example, were 0.7 and 0.3. So in our equation, P would equal 0.7, Q equals 0.3. Let's plug those numbers into this Hardy-Weinberg equation and just see what comes out at the bottom. So if we plug those numbers for P and Q into the Hardy-Weinberg equation and then grind through all the math, what comes out at the bottom is this: 0.49 plus 0.42 plus 0.09 equals 1. You should recognize those as being the three genotype frequencies that we had just calculated a few minutes ago. So this is just simply a demonstration that using the Hardy-Weinberg equation, if we know the allele frequencies, we can calculate the genotype frequencies in a population. And also, the opposite applies. If we know the genotype frequencies, we can calculate the allele frequencies. You will be expected to have memorized the Hardy-Weinberg equation for your exam, but just in case you forget it, this slide shows a way that you might generate that equation for yourself as a reminder. You could set up P and Q, the two allele frequencies, on both sides of a Punnett square. And if you do that and then fill in the boxes you would find PP, which is P squared. You would find P squared plus 2PQ plus Q squared. It all has to add up to 1. Equals 1 because it all has to add up to 100%. So this could be used to generate that equation if you forget it. The way this equation becomes most important for your exam is in its application to autosomal recessive diseases. Most of the test questions that I'm aware of on the Step 1 licensure exam apply this equation to autosomal recessive diseases such as cystic fibrosis, Tay-Sachs disease, or sickle cell disease. And here is the key to solving the kinds of mathematical problems that they will ask you to do on the exam for these kinds of diseases. For the autosomal recessive diseases, P and Q represent the two allele frequencies for some disease-causing gene. P, by convention, represents the allele frequency for the normal allele. Q represents the allele frequency for the mutant allele. Remember that the three parts on the left-hand side of the Hardy-Weinberg equation represent the three genotype frequencies. So in this equation, P squared represents the genotype frequency for the homozygous normal individuals. 2PQ represents the frequency of carriers, heterozygous carriers in the population. Q squared represents the frequency of homozygous mutant or affected individuals in the population. So, on your exam, I would expect you to see one or more of three possible questions. One possible question they might ask about this is simply, do you know what the different parts of the equation mean? For example, there might be a question that describes cystic fibrosis in a population, for example, and they might simply ask, what part of the Hardy-Weinberg equation is used to refer to carriers for cystic fibrosis? The answer to that, of course, would be 2PQ. A second way of asking a question about this is to give you the prevalence of a disease in a population, that is, the frequency of the affected individuals, and ask you to calculate the carrier frequency in a population. And the third kind of question that you may see is that they will give you the carrier frequency, 2pq, and ask you to calculate the fraction of a population that is affected by the disease, q squared. These are very common questions, and I would expect most of you to get at least one of these kinds of questions. Let's look at a couple of examples. In this first example, we're going to look at how we can apply this Hardy-Weinberg equation to the disease PKU, phenylketonuria. In the stem of the test question, they may tell you that the prevalence of PKU in a population is 1 in 10,000 live births. That means 1 in every 10,000 babies born has the disease PKU. The test question will ask, what is the carrier frequency? What is the carrier frequency for PKU in this population? So in this example, prevalence of PKU is 1 in 10,000 births. That tells you the frequency of the homozygous mutant people in the population, which you will remember is q squared. So in this, they have given us q squared, which is 1 divided by 10,000. They are asking us to calculate the carrier frequency, which is 2pq. So we know q squared, we want to calculate 2pq. Since we know that q squared is 1 over 10,000, we can determine q by determining the square root of 1 over 10,000. The square root of that is 1 over 100, or 0.01. Some of these test questions will be expressed or given to you in fractions, and some will be given in decimals. So you need to be able to do the math either way. So we've been able to calculate Q. We need to figure out what 2pq is. And one way of doing that would be simply to determine P by subtracting Q from 1. That is, by subtracting Q 0.01 from 1, we could determine that the value for P is 0.99, and then we could calculate 2pq by multiplying together 2 times 0.99 times 0.01 to get our answer. But there's a much simpler way that is better for your exam. Because the diseases that we are studying are relatively rare, P will be very close to 1. That is, in this particular example for PKU, P is 0.99. For other diseases, P might be 0.98 or 0.97, but it's always very close to 1. So for purposes of doing a fast and accurate enough calculation, we can simply round off P to 1. And if we do that, then the carrier frequency becomes 2q, and in this particular example, 2q would be 2 times 1 over 100, which is 1 over 50, or 0.02. So the carrier frequency for PKU is 1 out of every 150, or a frequency of 0.02. So note, in this example, they gave us the prevalence of the disease in the population, Q squared, and asked us to calculate the carrier frequency, 2pq. Some test questions might ask the opposite of that. That is, they may give you the carrier frequency and ask you to calculate the prevalence. And that is the case with this next example involving Tay-Sachs disease. At the top of this screen is the information that they may give you on the exam in the question. For example, they may tell you in a test question that 1 in every 30 Ashkenazi Jews is a carrier for Tay-Sachs disease. And then the test question would be, what is the prevalence of Tay-Sachs disease in the Ashkenazi Jewish population? Another way of asking exactly the same question would be, if 2 random Ashkenazi Jews got married and had a child, what would be the probability that their child would be affected by Tay-Sachs disease? It's the same question. So notice, 1 in every 30 Ashkenazi Jews is a carrier. 1 in 30 represents 2pq in terms of the Hardy-Weinberg equation. So we know that 2pq is 1 over 30, and we want to calculate q squared, the fraction of the population affected by the disease. Again, this is a relatively rare disease, and so we can simplify the equation by because p will be approximately equal to 1. And if we round off p to 1, then the carrier frequency will become 2q. So that 2q is 1 over 30. Q will be 1 over 60. And q squared then would be 1 over 60 squared, or 1 over 3600. So that the prevalence of Tay-Sachs disease in this population would be 1 out of 3,600. 1 out of every 3,600 Ashkenazi Jewish babies would be expected to have Tay-Sachs disease, or if 2 random Ashkenazi Jews got married and had a child, the probability that their child would have Tay-Sachs disease is 1 divided by 3,600. Now, most commonly, the Hardy-Weinberg equation on your exam would be expected to be applied to autosomal recessive disorders. And those were the examples we've already looked at. But technically, the Hardy-Weinberg equation applies to all kinds of disorders. It can apply to dominant disorders as well as X-linked recessive disorders. For dominant disorders, you have to remember that both the heterozygotes as well as homozygous mutant people will be affected by the disease. For X-linked disorders, the calculation becomes slightly different because males only have 1 X chromosome while females have 2 X chromosomes. The Hardy-Weinberg equation only applies when an individual or when a population has 2 copies of each gene. And that's true for the vast majority of the autosomal diseases, because all individuals have 2 copies of the autosomes. However, with the X chromosomes, only females have 2 copies of the X chromosome, so we can apply the Hardy-Weinberg equation to females for X-linked diseases, but we cannot apply this equation completely to males, since males only have 1 X chromosome. Now, this actually makes the calculation for X-linked diseases a little bit easier, because since a male only has 1 X chromosome, if he inherits a mutant X chromosome, he will have the disease. And so therefore, simply by counting the number of affected males in a population, we can directly determine Q. That is, the prevalence of an X-linked disorder in males gives us the allele frequency for that mutation. An example would be hemophilia A. If 1 in every 10,000 males has the disease hemophilia A, that directly gives us Q, the allele frequency for that mutation in the population. That allele frequency Q will be the same in both males and females, so that now that we have determined Q by counting males, we can now go back and apply the full Hardy-Weinberg equation to make predictions about females in this population. The number of affected females would be Q squared, which is 1 divided by 10,000 squared, that is 1 divided by 10 to the 8th, 1 divided by 10 to the 4th squared, or 1 divided by 10 to the 8th. So the fraction of affected females will be 1 in every 10 to the 8th people. That is simply a mathematical description of why in the pedigrees for X-linked recessive disorders, we essentially only find affected males in such a pedigree. It is possible for a female to inherit an X-linked recessive disorder, but in order for a female to have an X-linked recessive disease, she must inherit 2 mutant X chromosomes, and that is simply a very rare event compared with the likelihood that a male will inherit 1 single mutant X chromosome. We can also use the Hardy-Weinberg equation then to calculate the carrier frequency in females for an X-linked disease. The female carrier frequency would be 2pq. Again, we will round off p to 1 to simplify the equation so that the carrier frequency will be 2q. In this case, 2 divided by 10,000, that is 2 divided by 10 to the 4th power, or 1 in 5,000. So for hemophilia A, approximately 1 in every 5,000 females will be a carrier for this disease. The last section in Chapter 2 is a section that describes some factors that cause genetic variation in populations. Again, these are some things that can cause some complications or possibly mess up some of your calculations. Let's look at some of these other factors involved in this kind of variation in populations. One factor is new mutations. New mutations are the ultimate source of new genetic variations. Mutations are what led to all the genetic diseases that we have in the world today. If a new mutation were to occur, that theoretically at least would mess up your calculations involving the Hardy-Weinberg equation. When we did our calculations a few minutes ago, in our examples, we assumed that the mutations for whatever disease we were talking about, we assumed that all those mutations were already existing in the population and they were simply being randomly inherited in that large population. If a new mutation occurred, that would increase the value for Q and would change your calculation. However, new mutations occur at such a low frequency, such a small rate, that my advice is to forget about new mutations when you're doing Hardy-Weinberg equations, because those new mutations occur so infrequently they will not really change the values for your calculations. Another factor involved with variation in populations is natural selection, or survival of the fittest. Natural selection is what has led us to the particular allele frequencies that we have today. Through natural selection in all those populations of humans before us, there has been, for most cases, a natural selection against most mutations, most disease-causing mutations, because most disease-causing mutations cause problems for the individual, caused a decreased survival, and those mutations tend to be eliminated from the population. However, there is a very well-known example of a situation in which natural selection has actually worked in favor of a particular disease-causing mutation. And because that example is so well known, you should be sure that you know it for your exam. And this example involves the sickle cell mutation and the malaria parasite. The idea is this. The malaria parasite has to undergo part of its life cycle inside the red blood cells of the patient. However, in those parts of the world, Africa and the Mediterranean in particular, excuse me, in those parts of the world in which malaria has been a problem over many generations, it turns out that carriers for sickle cell disease are protected against malaria. It turns out that the malaria parasite cannot complete its life cycle properly inside the red blood cells of carriers, heterozygous carriers for sickle cell disease. That is, we refer to those heterozygous carriers as having sickle cell trait. They're heterozygous carriers for sickle cell disease. So because the malaria parasite cannot complete its life cycle in the red blood cells of those people, it turns out that carriers for sickle cell disease are protected against the effects of malaria, and so that over those generations before us in those parts of the world, there's been a selection in favor of the sickle cell mutation because homozygous normal people have tended to die of malaria, whereas heterozygous carriers for sickle cell disease have tended to survive malaria. That is why the sickle cell mutation is so common today. In the United States, approximately 1 out of every 10 African Americans is a carrier for sickle cell disease. And that means that roughly 1 out of every 400 African American babies will be born with sickle cell disease. Those are very high numbers for such a serious disease. The reason the numbers are so high is because of that natural selection in favor of the sickle cell mutation in parts of the world such as Africa. In fact, in some parts of Africa today, as many as 1 out of every 3 people is a carrier for sickle cell disease. Another complication is genetic drift. Genetic drift refers to gene frequency or allele frequency changes caused by small population sizes. These genetic drift is an effect that we might see, for example, in a small isolated population, perhaps a population isolated on some small island somewhere, or it can also occur in small populations that tend to intermarry just within their own relatively small group, the Amish, for example. And the idea here is that the calculations that we did as examples using the Hardy-Weinberg equation, those equations are based on statistics and probability, mathematical calculations involving statistics and probability. Statistics and probability really only work when you are dealing with large numbers. For example, if I were to flip a coin and ask you how many times I would get heads or how many times I would get tails, you would say that I should get 50% heads and 50% tails. If I flipped that coin 10,000 times, your answer would be pretty close to correct. That is, I might not get exactly 5,000 heads and 5,000 tails, but we would get somewhere in the vicinity of those numbers. However, suppose I only flip that coin 4 times. If I only flip the coin 4 times, then I might get 2 heads and 2 tails, but I also might get 3 heads and 1 tail, or 4 heads and no tails at all. Because of small numbers, statistics and probability does not work in small numbers, whether you're flipping a coin or whether you're dealing with genetics in a small population. So that if we have a small isolated population on an island, for example, that small number of people, because they are having only a small number of children, the children that they have might not match the predictions that you would make based on the Hardy-Weinberg equation. And so, in small isolated populations, you can have a tremendous increase in the frequency of a mutation in a population, or a tremendous decrease in the frequency of a mutation in the population in one generation, just simply due to variations to statistics by small population sizes. Some examples of when this might occur in genetics are such genetic drift following a founder effect or following new mutations. These are indicated in the next slide. The pedigree at the top of this slide is illustrating the founder effect. The founder effect occurs if a particular person moves into an area or founds a particular population in some area. If that person who founds that population carries a mutation, when that person passes the mutation along to the children, because he has a relatively small number of children, he may accidentally have more children than you would predict by chance alone who have received the mutation from him. And so, as that population then expands, what has happened is that the allele frequency for that mutation has increased in that population, and so the generations after this would have a higher frequency of this mutation than other populations in other parts of the world. This same sort of thing can also happen as a result of a new mutation in a family, which is illustrated there in the pedigree at the bottom of this slide. What has happened here is that a new mutation has occurred, and as this person then has children, just simply due to chance alone, more children than you would predict using the Hardy-Weinberg equation have developed the disease, have inherited the mutation, and so genetic drift has occurred in that small population, increasing the allele frequency for the mutation in that small population. Another complication is gene flow. Gene flow refers to the exchange or movement of genes between previously separated populations. A good example involves sickle cell disease in the United States compared with sickle cell disease in Africa. We mentioned a few minutes ago that the sickle cell mutation is pretty common in the United States. In the black population. The sickle cell mutation is common in the black population in the United States. However, it's not very common in the white population in the United States. However, nowadays we have lots and lots of intermarriages between American blacks and whites, and what we would expect to see over generations is the movement back and forth between those two previously separated populations of genes, and we would expect the frequency of the sickle cell mutation to decrease in the American black population. We would expect its frequency to increase in the American white population over time. Gene flow is the exchange of genes between previously separated populations. Another complication for trying to do calculations involving population genetics is consanguinity. This slide shows an example of a pedigree in which consanguinity has occurred. In this particular example, two second cousins have gotten married and are about to have children. The complication here results from the fact that when we did our Hardy-Weinberg equation calculations earlier, we assumed random mating in the population. It's one of the assumptions that Hardy and Weinberg made. However, if you have consanguinity, you do not really have a random mating anymore. If related individuals marry and have children, then because they have common ancestors and share a fraction of their genes, they are more likely to both have copies of the same mutation compared with two random individuals in the population. One of the topics related to this is the coefficient of relationship, that is the fraction of shared genes between related individuals. Such calculations can be done in the following way. Brothers and sisters share 1/2 of their genes with each other, and each parent shares 1/2 of his or her genes with each child. So the relationship, the coefficient of relationship between parent and child, for example, is 1/2. And between siblings, brothers and sisters, it's 1/2. So for example, in this particular pedigree, in which we have consanguinity involving 2 first cousins, we can determine the fraction of genes that those 2 first cousins share by the fact by multiplying the fraction of genes that they share at each generation. That is, at the top of the screen, there was the siblings shared 1/2 of their genes, and then the male in this relationship got another 1/2 of those genes from his parents. The female got another 1/2 of the genes from her parents. So we can determine that they share 1/2 times 1/2 times 1/2, or 1/8 of their genes in common. The fact that they have 1/2 of their the fact that they have 1/8 of their genes in common means that they are more likely than a random couple from the population to have a child with some genetic disease. People are still undecided about the exact factor that quantitation of the factor that this has in predicting the frequency of diseases, but the number that I've seen most commonly is that if 2 first cousins marry and have a child, they are about twice as likely to have a child with some genetic problem compared with a random mating in the population. And the risk goes up if the relationship is even closer. For example, suppose you had an uncle and a niece having a child together. An uncle and a niece would share 1/4 of their genes and would be even more likely to have a child with a problem. Suppose you had a brother and sister or father and daughter having a child together. We don't like to talk about that situation very much, but it does happen. A brother and sister or father and daughter would share 1/2 of their genes, and so they would be even more likely to have a child with some kind of genetic problem compared with random matings in the population. This effect of consanguinity is at least one of the reasons why marriages between related individuals are discouraged, although in some cultures and some parts of the world it is relatively common for first cousins to marry and have children. It does put them at a somewhat higher risk for having children with genetic diseases, but it is common in those cultures. So in this chapter, in population genetics, I believe that the highest yield material in this chapter, the part that is most likely to be on your exam, is the Hardy-Weinberg equation and the use of it in making the kinds of calculations that we talked about. I would guarantee you that most, maybe all of you, will have to do those kinds of calculations. Be sure that you know the Hardy-Weinberg equation and how to do these calculations.